Hello, this is Dr. Jose Kudakasseril. I am going to talk to you today about the tools of genetic engineering. We need two different tools in biotechnology or genetic engineering. One is DNA modifying enzymes. Three enzymes are required. One is restriction endonuclease that cuts DNA. Second is DNA ligase that joins DNA. Third is DNA polymerase that replicates DNA. Second tool is vectors. First, let us talk about restriction endonucleases. They are also called molecular scissors or chemical scalpels. They were discovered by Nathan Arbor and Smith, who got Nobel Prize for this discovery. These enzymes cut DNA at specific locations. They are called recognition sequences. They are produced by bacteria to cut and inactivate the DNA of viruses. They do not cut bacterial DNA because the recognition sequence is methylated in bacteria. These recognition sequences are palindromes. A palindrome is a word or a sentence which can be read in both directions. Example, Malayalam, a man, a plan, a canal, Panama. The recognition sequence of EcoR1, which is an enzyme obtained from E. coli, is GAATTC. Now let us talk about the naming of the enzyme. For example, in EcoR1, E stands for Escherichia, which is the genus. CO stands for coli, which is a species. R stands for the strain, which is RY13. And 1 stands for the first identified restriction endonuclease in E. coli. These enzymes are of three types. Type 1, which cut DNA approximately 1000 nitrogen bases away from the recognition sequence. Type 2, which cut DNA at the recognition sequence. Because of their specificity, they are used in genetic engineering. EcoR1 is an example of a type 2 enzyme. Type 3, which cut DNA approximately 25 nitrogen bases away from the recognition sequence. Type 2 enzymes are again divided into two. Those that produce sticky ends, example EcoR1, and those that produce blunt ends, example SMA1. In the case of EcoR1, it cuts at the specific location shown by arrows, resulting in the production of sticky ends, also called cohesive ends or overlapping ends. In the case of SMA1, which is obtained from Serratia marcescens, it cuts the DNA right in the middle. So the ends of the DNA has blunt ends. The first restriction endonuclease to be discovered was HAND2 from Haemophilus influenzae. HAND2 always cuts DNA at a specific sequence of 6 base pairs. The second enzyme used in genetic engineering is DNA ligase. It joins pieces of DNA using energy from ATP. It was discovered by Esther Lederberg. It is present in all bacteria. The third enzyme is DNA polymerase that replicates DNA. It was discovered by Arthur Kornberg. Taq polymerase, which is a DNA polymerase obtained from Bacterium thermus aquaticus, is used in PCR or polymerase chain reaction, which I will talk to you in the next class. Thermus aquaticus is found in hot springs. Therefore, this enzyme can withstand high temperature. So, we have discussed three enzymes used in genetic engineering: restriction endonuclease that cuts DNA, DNA ligase that joins DNA, and DNA polymerase that replicates DNA. I have published some books for students appearing for the NEET exam. One is Handbook of Biology, Volume 1, which describes all the chapters in class 11, and Volume 2, which describes all the chapters in class 12. And also I have published a book, Biology in a Nutshell, which includes all the chapters. These books are available on Amazon. I am giving the link below. In the next class, I will talk to you about vectors and their use in genetic engineering. Thank you for listening to my video.